So today I'm going to talk about a different aspect of NMR spectroscopy. So a lot of you have probably seen me talking about building probes, and today we're going to talk about the other end of the process, which is sample preparation. So I'm going to go through how do we pick an interesting protein target for structural biology. And before I do that, I want to give a little shout out or a little advertisement for some work that my group is doing. We're really into 3D printing probe parts and trying to make instrument building and testing easier for everyone. And we're making our designs freely available as we get things done. And Jessica Keltz and Jose Uribe in my group are working on that. And I hope that one or both of them will give a future iteration of this seminar series. So I just want to remind you to look for that. Okay, so let's talk about where little proteins come from. That's the background that we need to get started talking about how to choose protein systems from genome or transcriptome data. So in a eukaryote, the source code of the organism is in the DNA. And that's all locked up in the nucleus, and all the DNA replication and also the transcription, which takes it to RNA, happens in the nucleus. And then the mRNAs come out of the nucleus into the cytosol, and they have, mRNAs have some special features that allow the ribosomes to recognize them. There's a 5' cap, and then there's also a poly A tail that tells the cell that this is a transcript. And most human genes or most eukaryotic genes are monocistronic, which means you have one gene and that makes a single transcript, maybe with some splice variants, and then that makes a single protein. And that makes it pretty easy to move back and forth between DNA and RNA and protein sequences, which is pretty convenient. Although one thing that you will notice is that structural biologists get really sloppy about referring to the DNA and the RNA and the protein as though it's all the same thing. And that can be confusing if you're not used to it. But I want to point out that if we're talking about figuring out which proteins are doing some work in a cell, we can read this at any one of these three levels. So we can look at the source code. So that's the DNA, and that is the whole repertoire of every protein that organism could make. And then the transcriptome is everything that's being expressed, maybe in a certain tissue, maybe at a particular time, but that's what's actually being made. And then the proteome looks at what proteins are actually present. And each of these things could give you different answers depending on, so it's important to know what you're looking for. Just because something is in the genome doesn't mean that it's ever transcribed, and then proteins have different lifetimes. And of course they have different splice variants. So there are differences between the mRNA sequence and what actually turns into protein. But if we're just looking for interesting targets for structural biology or for chemical biology, we can make use of information at all these levels. And that's what I'm going to talk a little bit about today. So I told you about where proteins come from in general. Where do our particular enzyme targets come from in my lab? We have a couple different projects with this. We're looking at extremophiles that grow in the cold. But today I'm mostly going to talk about proteins that come from this plant, Drosera capensis, which is the Cape sundew, and it's the Cape of South Africa. So I actually went and visited it in South Africa, and it's tiny. This is the thing that I'm pointing to in that picture. But fortunately, we don't have to go out in the field to get them. We have a whole forest of them in my lab. And I want to point out that this whole project is a collaboration with Carter Butts. He does the genome assembly and some of the computational work. So why a carnivorous plant? Carnivorous plants are amazing biochemists. So when we eat food, we chew it up. And so the food gets disrupted mechanically, and there are already enzymes in the saliva. And then it goes to the stomach, which is a closed up area that's very low pH, and it's protected from other organisms in the environment. The carnivorous plant has none of this. It has to do its digestion right out in the open on a leaf. And it's in competition with all the bacteria and fungus that's in the environment. And all it has to do this is amazing enzymes. And so that's why we're really interested in this organism as a target for enzymes. And honestly, it's fun, and there are good reasons to look at these, but you can get really exciting biological targets for almost anything. I mean, this is one of my favorite things about biology, actually. You know, a literal pile of garbage has, you know, amazing molecules that you can work on for your whole career. But we've chosen to look at the ones from this plant. So in order to do this, we had to sequence the genome, and I extracted the DNA from this plant myself, which was quite an adventure. The protocols that you find for, you know, that turn out to be relevant for this, they use the term recalcitrant plant for plants that are hard to extract the DNA. And the reason that they're recalcitrant is, you know, there are several. They have a lot of pigments in the leaves that have polyphenolic compounds, and they derivatize the DNA and make it impossible to read. And then also a carnivorous plant has nucleases all over it. And so it's really difficult to, you know, to get the DNA isolated in a form where you can read it. But that is a story for another day. So the first time through this with the genome, I did the extraction myself, which was, you know, it was a lot of fun. And then when we did the transcriptome later, I actually kind of failed at that, and Gemma Takahashi and Omar Akbari in my lab have done it. Okay, so skipping over all the things about how you assemble a genome, we can talk about that later if anybody wants to ask me. I want to point out some of the quality control metrics that we use for a genome. So one is how much of the DNA did you read? So, you know, we extract the DNA from the cells, we read it, in this case, using Illumina technology, and then we can see, you know, how much do we have? And from this particular organism, you know, we expect about 300 million base pairs, and we got about 90% of that in our genome. So that's not so bad. And then we can also look at whether it has all of the core genes that we would expect for a eukaryote. And so that's where we can look at these gene ontology codes. And that's just a classification of, you know, what different proteins do. And so again, you know, we're using gene and protein kind of interchangeably here, like all of these things refer to the activity of the protein. And we can get kind of a sanity check for how good our genome is by just looking at how well it covers some of the, you know, the essential functions of a eukaryote. And again, you know, there's a list of about 250 core genes that, you know, everything kind of has to have to be, you know, to be a viable organism. And we got 100% matches to about 90% of them, and partial matches to essentially all. So that tells us that we, you know, we do have the source code for this plant. So now what? So this, you know, this is, it's hard to figure out what to do with so much data. So, you know, a typical eukaryote has tens of thousands of genes. We can't make them all. We definitely can't do NMR on them all. So what are we going to do? The first thing I looked for in this genome was a particular protease. And I did that because in 1875, Darwin wrote a book about carnivorous plants. You know, he was a prolific guy with a lot of interests, but one of the things that he did is write a whole book about carnivorous plants. And he said that this plant in particular, the sundew, has a ferment that's similar to mammalian pepsin. So it was 1875. He didn't know the word protease, but he was already saying that the plant had the same kind of molecule as protease, and that's what was enabling it to do the digestion. So that was the first thing I looked for. I did a blast search against mammalian pepsin, and I found all kinds of aspartic proteases that are similar in the plant, you know, which is kind of amazing. We're looking at this thing's digestive enzymes. It's very far away from a mammal on the evolutionary tree, and it does have these kinds of aspartic proteases that we're used to seeing in digestion. And I should point out, in this little tree, our plant is not, it's, you know, it's very special to me, but it's not that special in the sense that lots of its relatives have similar proteases. So in this tree, you can see, here's our little sticky leaves from the Drosera. Also, we looked at some from the Venus flytrap. Somebody had published a transcriptome. There's now a genome for that one too that another group published. And also the nepenthes, the Asian pitcher plants that are arboreal and hang down from trees, those have aspartic proteases that are similar, and even some other model plants. And the lesson here is that, like, yeah, carnivorous plants have amazing enzymes, but they have to use the starting materials that all plants have. So these things diversified from other maybe housekeeping enzymes or other things that were already present in plants. Plant carnivory has evolved something like 8 or 10 separate times, which I found amazing. I mean, there are all these carnivorous plants that, you know, some of them look pretty similar and they're not actually very close relatives. But these three are relatively close to each other. Okay, so now we have the source code. How do we know what's actually expressed in the cells when the plant is doing its business? And that's where Gemma Takahashi's experiment comes in. So she took one of our sundew plants in the lab and she treated some of the leaves with just water and some of them with jasmonic acid, which is a plant hormone that induces the digestive process and causes it to do its feeding behavior where the leaves curl up. And she did that to see which proteins get expressed or upregulated during the actual digestion. And that does work. So we see that, you know, with the leaves that were treated with water, nothing happens. The ones that were treated with jasmonic acid do curl up as though they caught an insect, even though there's nothing there. And the reason that we don't just feed it something is that then we would have insect RNA in our sample and it would cause a lot of problems. And we only want to see the proteins that are from the plant. So we do get a lot of genes that are differentially expressed. You get, you know, where you get either more or less in response to the feeding behavior. And notice that we have plenty of both. The blue ones are things that were expressed less than in the control, and the red ones that are things that are expressed more. And so if we look at what actually changes when we do that, we see things that make a lot of sense. So transcription factors, proteins that control the expression of other proteins, transporters. Transporters sound like a really good thing to have if you're a carnivorous plant. It has to you know, digest its prey and then bring all those nutrients inside the cell. And we also see some digestive enzymes, including cysteine proteases and aspartic proteases. And so remember, aspartic proteases are the ones that Darwin hypothesized were there. Let's look at some of the cysteine proteases first, just as an example. So there are a bunch of them that are upregulated, and I'm showing you predicted structures of a couple of them, just for example. But one thing that is going to come up as a theme later is this plant has a ton of cysteine proteases. And when I first started looking at this, I was so excited. Our plant has 44 cysteine proteases. It's really special because it's a carnivorous plant. Well, it turns out all plants have a lot of cysteine proteases. These are enzymes that are involved in defense and in resistance to environmental conditions that are tough. So it turns out that rice has about twice as many. So it's, you know, you get a lot of surprises when you start looking into these things. Okay, so once we have our genome and transcriptome data, then what? How do we pick targets? We have a huge number of proteins. And I'm going to go through two different strategies that you can use to pick targets out of this thing. And the first one is going for a target that you already know about. So this is the one that was given to us by Darwin. We're going to look for aspartic proteases. And so you get your transcriptome data, which looks like this. We have a whole bunch of sequences that, you know, that code for those proteins. And again, this was done by Gemma and Omar. And they have these tags here. And so this xloc is the identity of the particular sequence and tcons is the transcript. So this is just telling us where these things are located in our data. And it can be hard because we have different identifiers for these proteins in the genome than we do in the transcriptome, you know, just because of how the data are processed. And so one of the important steps is to go back through and correlate everything that we see in the transcriptome and find out where it is in the genome. And of course, along the way, that helps us make our genome assembly better. And that's something that, that Gemma and Omar are working on right now. So going back to Darwin's aspartic protease, we do see at least one aspartic protease that's very highly expressed during the feeding behavior. And so Omar pulled that out of the data and used a blast search to find proteins that are related to it. So this step is important because it enables us to pick reference sequences that we can use to figure out what's going on. Because, just having the sequence doesn't help you very much. All right, you've got a bunch of nucleotides, or you can translate it to amino acids. So what? How do we know what it actually does? So if you do a blast search using our search, or using our protein against the database of known proteins, we find that the closest relative to this aspartic protease is in the beet. So both Drosera and beets are in order Caryophyllales, which is a large plant order. And, the beet has no carnivorous tendencies as far as I know, but it is kind of the closest, very well characterized relative of this plant. So that's, it's kind of fun to see, where your reference sequences come from. However, that aspartic protease from the beet is not very well characterized itself. And so, we needed to go look for something that, that had more information about it. And so how about one from Arabidopsis thaliana? That's kind of the Drosophila of the plant world. A lot is known about it. There are very high quality genome assemblies and lots of experimental data. So then we looked for an Arabidopsis aspartic protease. Okay, so to be able to compare our protein sequence to other ones, we first need to translate it. So what I've been showing you up to now is nucleic acid data. You can see the G's and T's and C's and A's, and that doesn't help us so much with the protein properties, and so we need to translate it. There are some free tools that you can use to do this. And I want to say that, I'm going through the process a little bit quickly, but I did share with the organizers a list of free online resources that you can use to do these operations. And that's going to be posted in the comments in the YouTube video. So make sure to check that out. I tried to focus on things that are free and it's just on a website and you can go use it and it's pretty easy. There are more sophisticated bioinformatics tools that you can use if you get really into it, but you can go really far with these free online tools. So those are the ones that I collected there. Okay, so we have our nucleic acid sequence that we got from the transcriptome, and we need to translate it to protein. So here's the problem. Every codon that codes for an amino acid is 3 nucleotides, and you don't know where in the code it starts. So you have a transcript, here's your protein, but you don't know that you're actually getting it at the starting point of the protein, and you don't know which reading frame it's in. So if you have 3 bases per amino acid, you don't know which one is the beginning, and there are different reading frames that you can use. And so we have to try to figure out which one is happening. Okay, fortunately, we know what a start codon is. It's ATG or AUG in mRNA. And we have various options for stop codons that are used by eukaryotes, and we know what they are. And so we can translate our nucleic acid sequence to protein. And there are all kinds of different reading frames. How do you know which one is right? Usually it's pretty obvious when you put it in, because when you see a long unbroken sequence, between the start and stop without any gaps, probably that's the right one. None of these other reading frames have anything that's long enough to be a real protein. And it's almost always the case that you can tell pretty well. And so now we have our sequence. We can use it to compare to other protein sequences, and we can predict the structure using one of the structure prediction methods that's out there. There are various versions of Rosetta. There's iTassr, there's AlphaFold. We've used them all at different times. They're useful in terms of being able to see what you think the structure could look like. And so this is our highly expressed aspartic protease predicted using Rosetta Fold by Omar Akbari. So he's an undergrad chemistry major, and his undergrad project is he discovered a new enzyme, which is pretty cool. All right, so that's just one protein, and you could run with that. Honestly, you could take the whole thing and and go and start expressing that protein and use it for NMR. But there are some other things that we can do too. So one way to choose your target is just to say like, okay, which one is most highly upregulated in the plant? And that's a fine way to do it. But it's also useful to look around at, what are some other related things in there? Because as chemists and, biophysicists, we're not necessarily always interested in the thing that's the most abundant in the organism. In some sense, we don't need to be constrained by that. We're interested in things that we can use in the lab. And so it makes sense to look at some of the other sequences that are around. So this is going back to the genome sequence and looking at some alignments of the aspartic proteases that we found with some reference sequences. And here again, our reference sequences are pig pepsin, and then our aspartic protease from Arabidopsis thaliana that we found. And so when we align these sequences, of course, you can't see the details here, but it's color-coded in a way that shows you some important features. So the little red arrows are the active site residues. So those are the residues that actually do the chemistry and cut up the other proteins. And then we have some definite sequence regions that we can identify. So there's a signal sequence. This is what tells the cell that this protein needs to get exported, and it gets exported either into the vacuole in a normal plant protein, or in this case, outside into the digestive mucilage so that the plant can break down its prey. It also has a pro sequence, which is like sticky tape covering up the active site when the protein is initially expressed. And the reason that's there is that if you just made a protease inside the cell and let it run around unsupervised, it might cut up proteins in the cell that it's not supposed to. And so a lot of them are expressed with pro sequences that keep them covered up until it's time for the activity to start. And then we notice there's this region that's highlighted in green that is not in the pig protein, and it's only in some of our Drosera proteins. And that's a really interesting feature that it turns out that's what we took NMR spectra of. So I will get back to that in a little bit. Before I do that, I want to talk about how do you find your reference protein. So I mentioned that the beet protein wasn't good enough because there wasn't enough information about it. And I really wanted something that had been better studied in order to get a handle on what our new protein does. And to do that, I went to Uniprot. So this is another free website that you can look at. And it has mind-boggling amounts of information about every protein that has been studied. Of course, nothing's perfect. Sometimes you do have to go back to the literature to find important information, but there's a lot of stuff in the Uniprot database that can help you. All right, so here's our protein. It's called aspartic proteinase A1 from Arabidopsis. And some important features to look at here is it has an annotation score. And this one has 5 dots. That means it's really well studied. So it has experimental evidence at the protein level. And so these dots vary from, somebody sequenced a genome and it was predicted that this gene exists, to like somebody verified that it actually gets expressed at the mRNA level, to, all the way to the best rating, which is somebody has actually done experiments on the protein and we know that it exists and it's functional. We also have some notes about the function of this protein. It tells us that it breaks down storage proteins in the storage vacuoles. And so a lot of times that's in a seed in the context of a plant. It's breaking down protein that was stored for the developing embryo. And it also tells us where the active site residues are. And that's really valuable when we go to make those alignments and try to figure out where they are in our protein, because we're going to need to compare them. There's a lot of really useful information in Uniprot, and you end up learning some biology from looking at this. So it tells you that this protein is found in the vacuole, and it gives you a little diagram of the plant cell with the vacuole. That's kind of cool. And then if we look in more detail at the sequence analysis, we can see different sequence features. So I already pointed out the active site residues, but it also tells you that this thing has a signal peptide. I mentioned that's how the cell knows to export it from the cytoplasm. And it has that pro sequence that covers up the active site before it's ready to go. And then the rest of the protein, here it just says chain. That's the main functional domain of the protein. We also have some disulfide bonds, and it tells you where they are. There's a glycosylation. So all of these kinds of modifications that might be important to the protein function, the ones that are known, are going to be listed. And they will also have citations for where these things are published if you click through it. So there's our important sequence information. It also gives us some information about the different domains of the protein that have been identified. So a lot of times, proteins have different functionalities that are stuck together. And this one has a domain that is peptidase A1. That's our protease that serves a digestive function. And then it also has this other thing in the middle. So notice this is in the middle of that one. We have 82 to 503, and then we have 312 to 417, and it's called Saposin B type. So that's really unusual. It's often, we wouldn't have one domain that's inside another one. Like a lot of times they're kind of arranged like beads on a string sequentially. And so, that's something to go and look at further. And in order to compare this protein to other proteins and get the sequence alignments, we need to download the FASTA sequence, and that's also on that UniProt website. And it has a little download button. And so you can collect these things in a text file and start using them for alignments. So that's our reference sequence. That's our roadmap for figuring out what's going on with the new proteins. And we can align them using a tool called Clustal Omega. There are plenty of these tools. This is one that's free, and there's just a website. You can paste in your sequences and it doesn't take very long. And so once we get that, we can put in all of our sequences from the plant and see how well they align with our reference sequence. And sure enough, it turns out that we can see the active site residues. They're in the same place as they are in the reference. And then the disulfide bonds line up. That's what's labeled in yellow. And then a whole bunch of these proteins have this saposin sequence that's in the middle of the Arabidopsis protein. So we can also check our own proteins for signal sequences. There's a program called Signal P that finds signal peptides. And again, this is from 2016 when we first sequenced the genome. Signal P has improved a lot since then. It has all kinds of exciting features that can identify different things that in your protein. But here we're just looking at, does it have a signal peptide, yes or no? And so here you can paste in the sequence of your new protein and figure out, is it likely to have a signal sequence or not? And here these are the ones from the paper, so I'm only showing you examples that do have a signal peptide, but it'll tell you yes or no. And so if we're looking for digestive enzymes, if we find something that doesn't have a signal peptide, either it's something that stays inside the cell, in which case it's probably a housekeeping protein or something, it's not digestive, or else maybe the sequence is incomplete. And so we excluded things like that from the published data set because we're looking for digestive enzymes. All right, so is this useful? What did we learn? So the first thing that we did is predict the structures, and in this case we used Rosetta, and we were able to use the Rosetta structures to actually improve the annotation of this Arabidopsis protein that was in UniProt. So the positions of things like the pro sequence and the signal sequence are based on sometimes they're based on experimental data, but sometimes they're somebody's best guess by comparison to reference proteins. And if you align this Arabidopsis protein with mammalian pepsin and just look at the sequence, you get one prediction about where to cut off the pro sequence. And if you do that and model it, you get something that looks like this. So here the catalytic domain is in green and the pro sequence is pink. And so just by comparing the sequence to pig pepsin, we get something that has a little bit of pro sequence left over. And this was what was reported in UniProt. And if instead we do structure prediction and align our predicted structure with the experimental structure of pig pepsin, we get a little bit different place to cut the pro sequence. And now you can see those red aspartic acid residues sticking out there ready to cleave the other proteins, just like they are in the active pepsin. And this is pretty useful because there's a good chance that if you made this in your lab, it wouldn't work and you wouldn't know why. And so this approach of using structure prediction as an integral part of our target selection can help us predict some chemical activity and choose good targets. All right, so going back to Droserasin 1, our aspartic protease that we picked from the Drosera capensis. If we make these kind of Rosetta models, we can see some interesting structural features, that we already knew about from the sequence, but it helps to see in 3 dimensions where do they go. So here's the signal peptide. That's what's going to tell the cell to export it. It looks like a handle sticking out. You can really imagine how the secretion mechanism is going to grab that thing and drag it through the membrane. And then again, our pro sequence in pink is covering up the active residues that are shown in red here. And then look at this green thing. That's that little saposin sequence that was found in some of these plant peptides. It turns out that is called the plant-specific insert, and it's really interesting. It's like an enzyme within an enzyme. So if we look at all the things that get cut off from the Droserasin 1 and examine just the mature enzyme, here's what it looks like. So it's pretty similar to mammalian pepsin. It has a fairly large active site cleft. Lots of proteins can go in there and then they get chopped up by the active site. Here's the mechanism, which I'm not going to go into in great detail, but it's interesting to have it there just so we can understand what features are important. The close-up, though, shows you that you do have these 2 aspartic acid residues that are pointed at each other, ready to attack the backbones of the substrate proteins. Okay, so we think that the plant-specific insert gets cleaved off the protease in the mature enzyme that's active outside the cell. And I want to point out that I know this because of pioneering work by Ricky Yada, who's at UBC. He's worked on PSIs in the potato and also in that Arabidopsis protein that I mentioned and a few other ones. And his papers enabled me to figure out what's going on with this subsequence, which is really interesting. Okay, so during the maturation of this protein, the PSI gets cleaved, and then it goes off and has its own adventures. It makes this little 4-helix bundle that has 2 states. There's a closed one and an open one. And we think the open one is the active version. Oh yeah, so what does it do? It's an antimicrobial peptide. So when you express these proteins, you get a protease and an antimicrobial peptide all in one. And if we do structure prediction on that plant-specific insert by itself, we get a range of structures actually that's pretty interesting. So we get this open form, which shows up as a dimer because the crystal structures of it are dimers. Is that dimer biologically relevant? I don't know. I wonder if this open monomer binds to the membrane instead. That's something that our collaborators have done some MD simulations on. And then it also has this closed form, which is what is similar to saposin C. And I want to point out that this also looks a lot like other known antimicrobial peptides. So for example, here are some human defensins that have this similar kind of structure where we have this little compact thing that is held together by disulfide bonds. And I'm showing you this just to illustrate the process of how we can go back and forth between sequence information to structure prediction to looking at known structures that are in the literature to try to figure out what this brand new protein might be doing and whether it's worth doing experiments on. Okay, so we did make this PSI in my lab. This was done by Mark Sprague-Piercy, Jan Bierma, Natalia Kozliuk, and actually a whole bunch of people in the group. And they were able to make a bunch of this little peptide, and they showed that it is very, very stable. So if we do circular dichorism, that's a good marker of secondary structure in a protein. And it turns out that you can heat it up to 90 degrees. You can almost boil the water, and nothing happens to it. If you heat it up to 90 degrees in the presence of reducing agents, that's this dashed line, then at lower pH, eventually it starts to show some effects. But this thing is incredibly stable, which is good. That's a feature you want in an antimicrobial peptide. And we can also see experimentally that it interacts with lipids. So in this experiment, we're looking at dynamic light scattering data, which roughly gives us an indication of the size of the particles. And we have E. coli lipids that we're following over a period of time. So it starts with the purple curve, and then we take a bunch of time points up to 4.5 hours. That's the red one. And we see that the lipids alone mostly maintain the same size of the vesicles. Maybe there's a little bit of spreading out, but mostly nothing happens over a range of pHs. When we throw in the PSI from our plant enzyme, the result is really different. So above pH 5, nothing happens. But at pH 4 and 5, we start to see this really dramatic shift in the size of the vesicles. They get a lot bigger. And so that indicates that the membrane is getting disrupted. We're assembling these really large vesicles due to the presence of the PSI. And this is interesting because the pH of the digestive mucilage of the plant is about 5. So it's active in the pH range that we expect. And we did the same thing with yeast lipids. So we're looking at bacterial membranes and fungal membranes. And the same thing happens with yeast. In a way, it's even more dramatic because the yeast membrane vesicles stay together better on their own. And then we get a lot of disruption when we introduce the PSI at low pH. In order to figure out how this might be working in the membrane, our collaborator, Carter Butts, did an MD simulation of this. So this is the dimer, and you can see it sitting in the membrane, and it starts to make a water tunnel through that membrane. It's just moving around, and you start to see the water get pulled in. And so you could imagine how this could start to really disrupt bacterial membranes, and that this might be important for its antimicrobial function. And that's really interesting. It helps us generate a lot of hypotheses, but at this point, we don't know whether that's actually the mechanism of membrane disruption. And so, of course, the answer to how to figure that out is NMR. And so that's something that we're doing. And we've worked on this protein in collaboration with the the National High Field Magnet Lab in Florida. And the reason for that is that we want to try to maintain this thing at as close to physiological concentration as we can, and so it doesn't have a huge amount of peptide in the membrane. And so we used the series connected hybrid magnet that's 1.5 gigahertz, and so we were able to take some preliminary spectra of that. And we find that the PSI is very strongly bound to lipids. So if we just take some 1Ds, we can see that an N15 inept gives us nothing. Whereas if we do cross-polarization, then we see nice strong signal, both at 700 megahertz and at 1.5 gigahertz. Although at the higher field, we're getting more signal out of it for less sample. And so we've also tried some of the preliminary experiments that we need to start solving a structure. And this is something that we need to get back to, once we're able to travel again. But the preliminary results look pretty nice. So this is the first example for us where we went all the way from discovering something in the genome to taking NMR spectra of it. And I think that's, it's a lot of fun. It's really great to be able to choose our own targets at that level and really discover things that are new. Okay, so I told you about one strategy for finding a target, which is, picking something that you knew was already there. But that can be hard, and especially because it's difficult to make use of ever bigger data. And what I mean by that is genome and transcriptome sequencing has gotten really cheap. You can do lots of it. And so the count of, in terms of base pairs in the NCBI database, where all known DNA sequences are compiled, it doubles every 18 months. And recently, UniProt had about 2 to the 8th annotated protein sequences in it. And then there's a huge mismatch between this and the experimental data. So expressing a protein costs, a few $1,000s, and that's if everything works well. Sometimes it can be much more difficult. And then, as we know, making NMR samples and processing the data and all of that is really time consuming. And so we want to make sure that we're spending our time on the most interesting targets. And as I mentioned, that may not necessarily be the one that is highly upregulated in the organism. It might be something else. So as chemical biologists and people that are looking for tools for biotechnology, what can we do? And I think that structure prediction is really the answer. And it's not that structure prediction replaces the experimental structures. It doesn't, for a bunch of reasons. But it's a really great tool to enable us to pick the best targets that are worth spending our time on. And so, again, this whole project is a collaboration with Carter Butts and his group. And the idea is we're going from our DNA or RNA sequences through, discovering particular families of genes and then annotating the features, predicting structures en masse for a whole bunch of the proteins that we're interested in. And then structure equilibration is, basically some more advanced MD simulations beyond just the initial structure. It helps us get a more realistic picture of the protein. And then we can analyze those predicted structures to tell us which proteins are worth taking for experimental validation. So to give an example of where we did this, let's go back to the capensis cysteine proteases. So for the aspartic proteases, there were about 7, and it's cute, and they're all pretty similar. So, it doesn't matter so much which one we pick. For cysteine proteases, that is not true. There's 44 of them that we found from this plant that are brand new, and they're very different from each other in terms of sequence. So what are we going to do? These are, a few that are related. I just want to show you the overall structure. So again, we see the active site residues. Those are the arrows. Anything that did not have the active site residues got thrown out. And then it has a signal sequence in peach and a pro sequence in pink. And our reference sequence in this case is papain, which is a cysteine protease from papaya, and it's made in the fruit. And the point of it in the context of the plant is to keep insects from eating it because it's supposed to be dispersed by animals. And it has a really recognizable active site cleft and a catalytic triad that, that we're going to look for in our new sequences. So when we go to do our structure prediction, we don't just use Rosetta or AlphaFold or whatever and stop there. We also do a process that we call in silico maturation, which is that we try to replicate the important post-translational modifications the best we can in silico, and then also equilibrate it using MD. And so what does that mean? It means we put in the disulfide bonds, and we also minimize it so that we get a more realistic looking active site conformation. And one question that people like to ask is, how well does this work? And it's a fair question. It turns out that we were really lucky while one of these papers was under review, and somebody solved the crystal structure of a protein from the Venus flytrap that we were working on. And so that structure wasn't available when we were doing the predictions. And it happened while our paper was being reviewed. Here's the comparison. So the gray one is our predicted structure, and the green one is the crystal structure that was solved a little bit later. It's amazing. It looks like magic. It's not magic. The reason this works so well is that there are lots of enzymes in this class that have been previously solved. And so the structure predictors have a lot of information to work with. So can we find new functionality? So we have this basic papain scaffold that most of these proteins share, but we can also see if we look at them that actually a bunch of them have special features. So this is one that shown in orange that I found from this plant that actually has this big kind of loopy thing with a couple of helices that's sticking out of the active site cleft and partially blocking it. And in other cysteine proteases, such as this one that's shown in light blue and this other one that's shown in dark blue, there are these little kind of turns that stick out near the active site cleft, and those actually modulate activity. So if those blue things can modulate activity, this big orange loop, must be able to affect the activity of the protein. And so we hypothesized that maybe it's a switch, maybe it has different conformations at different pHs. That's not something we know, though. We need to go and look at it experimentally. This protein also has some differences in the active site that show that are different from the standard plant aspartic proteases. But I did find an active site in a viral protease that's similar to this one. So as far as we know, probably it's active, but we still need to do experimental verification. We've also found some new types of granulin domains. So these are these extra C-terminal domains that are on the papain scaffold. And what they do in the plant is they make these proteins all stick together into little protease nanoparticles. And the reason they do that is that these are seed storage protein proteases. So in the developing seed, all these proteases are stuck together. They're ready to release the storage proteins when the seed starts growing. And so they pack into these little nanoparticles. And then when they get hydrated, when the seed starts growing, they release and chop up all the storage protein. So that sounds useful, right? Little protease nanoparticles. There are things we could do with that in the lab. And I want to point out that there were two types of granulin domain known from these sorts of proteins in the literature, and we discovered two more just from the ones in this data set. So there's a lot you can do if you go through this kind of analysis. And the last thing I want to mention is that we can do some other analyses of these structures. So for these predicted structures. So for example, we can make protein structure networks. And this methodology, the ones that we use were first pioneered by Valerie Daggett. And the network that she uses has these definitions of the moieties. So we have particular chemical functional groups that they don't correspond to individual amino acids. Instead, they're corresponding to chemical functionality. So the carbonyl group of this aspartic acid is a different moiety than the hydrophobic part of it, kind of the stem. And then to make the network, you just look at which moieties are within van der Waals contact of each other. And so each chemical group is a node. And so that's going to be a dot in our network. And then two of them are tied together if they're close enough to interact. And so we can get this network representation of our protein structure. And so if we do that and we use principal component analysis on our set of cysteine proteases, one thing that's kind of satisfying is papain is in the middle, but it makes this interesting kind of tripartite structure that is characterized by these different protein networks that look pretty different if you plot them. And so if you want to make sure that you get the most diverse range of cysteine protease functionality from this data set, what do? You make this one and this one and this one. And that is something that we're doing, but I can't tell you how the results are yet because we're not finished. Okay, so I'm almost done, but I want to mention briefly that since the pandemic started, we've been using a modified version of this pipeline to look at which variants of the SARS-CoV-2 main protease are likely to impact its function. So that's something that we've been working on for the last year and a half or so. And we have a whole bunch of MD trajectories of different variants. And we have seen that the protein is, it does seem to be evolving. There are some significant differences in its structural properties. And that is what I wanted to say for now. I need to thank my group and collaborators. So first, Carter Butts, this whole project is a collaboration with him. He did the genome assembly and the MD simulations and all of that stuff. And then the group members that have worked on this, I mentioned Jan Bierma, Natalia Kozeljuk, and Mark Sprague-Piercy. They graduated earlier and really got some of this stuff started. And then more recently, Gemma Takahashi and Omar Akbari, and then also Marquise Crosby have been working on these, making these proteins and getting experimental evidence for them. And thank you for your attention. I'm happy to take questions.
Thank you so much, Rachel, for this fantastic seminar. This is, a great tutorial to walk us through the how kind of what is the structural functionality relationship of proteins and how structural prediction could help us to better understand the functionalities of proteins. It's really great. So if you have any questions from the audiences, please feel free to type them in the Q&A channel. And while we're kind of waiting for questions, maybe let me make a very important announcement. The next lecture series, which will be on February 8th, will be given by Professor Geoffrey Bodenhausen on the topic of singlet NMR. And the tentative topic right now is long-lived states, some unexpected applications. So we're really looking forward to Geoffrey's seminar on February 8th. So please stay tuned. I haven't really see any questions raised yet in the channel, which kind of is a good indication that you really walk through everything progressively in great detail. So that's a good thing. I do have a question about kind of your general opinion of how NMR technologies could kind of further play a role in this entire, let's call it a system biology framework at different levels, at the genomics, proteomics, transcriptomics, or even metabolomics. I mean, I had a little bit experience with the metabolomics, which I can definitely see NMR is playing a increasingly important role there. And from your kind of a seminar, I think I can definitely also see the importance of NMR in trying to kind of understand, or especially experimentally decipher how protein works on the structural side, on the dynamics. What's your opinion of NMR, the potential roles NMR could play to further advance this kind of overall framework?
Yeah, I think in a way it's almost the most important thing, and having good structure prediction really helps us use it more effectively, because just having a structure isn't so exciting. I mean, I like solving structures. It's really fun to do, but okay, you get a snapshot. Now what? So, what we're really interested in with enzymes is, how does it interact with other molecules? How does it grapple the substrate? And, tell you what, even for really well characterized proteases like this, that is completely not known. And it's something that we can address with NMR. And it's easier to address now that we have, a better idea about, the structure of the protein. And things like these structure prediction programs make it also easier to refine our NMR structures. And so at this point, it's easier to get more structures per amount of NMR time. And that enables us to tackle these kinds of chemical problems and problems of dynamics. And for example, how does the protein interact with the membrane, and these kinds of functional questions that are more interesting.
Yeah, that's great. We got one question from Amrit, which is one of our organizers. He asks, how long did the nitrogen 15 CP experiments on the 700 megahertz versus 1.5 gigahertz spectrometers take? So how long does the experiment take on different fields? And could you also comment on the gains in sensitivity?
Okay, so sorry, I don't remember exactly how long those spectra took. The protein is labeled though, so not long. I mean, these were not heroic natural abundance experiments. Yeah, there was a little bit of loss in resolution, but it wasn't as bad as I expected. Actually, it was it was pretty decent. And these spectra that I showed were really our first try, and then we kind of switched gears during the pandemic. And so the plan is to go back to that and hopefully optimize the sample preparation a little bit more. So I think that one thing that is possibly hurting us more than homogeneity issues is that we don't have the lipid to protein ratio worked out perfectly yet. So I'm not sure that our sample is completely homogeneous, and that's something that's definitely on the list to look at.
Yeah, great. I mean, you already commented on the resolution as well. That I was going to hold that question later, but yeah, that's great. We do have another additional questions in the Q&A channel. Tom Osborne Pop asked, yeah, he said, great talk, thank you. What would you recommend for when your protein target doesn't have similar structures? For example, making a tool like Drosera less effective for initial structural prediction.
Yeah, that's an interesting question. Okay, so I didn't have time to talk about it, but we actually did publish a paper where we tried that. So we have a different class of enzymes where they only have between 20 and 30% sequence identity to known enzymes, and we used Drosera anyway. And basically, what happens is, my confidence in the structures goes down, but we can still use it to generate hypotheses. And so, so I think the answer is you just have to take it less seriously, and and not say we really know the protein looks like this, and we're going to develop more high-level hypotheses about how it grapples the substrate. Or I think you have to kind of take it a step back and say, now my hypothesis that I get from this is that this enzyme looks different from this other one, and the structural data will tell us that. So I think it's still useful. Also, these structure predictors have improved just phenomenally since we started doing this in 2016. So, with AlphaFold, and then the other competitors, Rosetta and iTaser have started using similar methods. They all work a lot better. So that's really great. It helps us a lot. Of course, they work really well for things where there are a lot of structures like that in the PDV. We can't get away from that problem. So again, I think the best way to look at it is just it's a tool that can help us develop hypotheses and help us pick the most interesting proteins to work on. But you still have to do the experiments at some point.
Yeah, great. Another question from an anonymous attendee asked, I guess it's pretty general, do we require a technique like DNP to overcome sensitivity issues? So
I would say, yeah, DNP is great. It really enables us to look at all kinds of proteins that otherwise would be too difficult, because you can't make enough of it or the concentration is really low. But do we require it? Not necessarily. It depends on what you're doing. So for some proteins, you can make plenty of labeled sample, and you get decent sensitivity without using DNP. But it's definitely another useful thing to have more tools in the toolbox.
And again, we got another question here. Can you explain why DAR has more side residues at the 32?
That's an interesting question. Okay, so what I would love to say is, we're seeing we're seeing more peaks because we have better sensitivity, but I'm not positive that it's not differences in the sample preparation. Like I said, this is kind of the proof of concept, and it's something that we need to go back and explore in more detail.